Welcome, everybody. We're on YouTube Live. Hope you've been able to tune in okay. I'm here with Dr. Anna Forsman, who has her program tonight on the Purple Martin Project. And I'll give a quick introduction. Dr. Forsman is a research scientist in the Department of Biology and the Genomics and Bioinformatics cluster at UCF. Prior to joining UCF in 2016, Dr. Forsman completed her PhD work in the Department of Ecology and Evolutionary Biology and the Lab of Ornithology at Cornell University. And we're big fans of the Cornell Lab of Ornithology. We use All About Birds all the time. They've been sponsors of the North Shore Birding Festival, so that's a wonderful place to have graduated from. Dr. Forsman is broadly interested in the relationships between wild birds and the microorganisms that live in, on, and around them, for example, in the gut microbiome. She's particularly interested in how bacterial communities in the environment interact with the avian immune system and how these interactions influence immune system development and function and overall bird health. At UCF, Dr. Forsman teaches courses in ornithology and genomics, which I'd like her to define what is. Before we get started, this is Orange Audubon Society, and that's our website, orangeaudubonfl.org. We have a big thing happening is that the North Shore Birding Festival is coming up December 3rd through 7th, much earlier than usual. And you can sign up for updates and follow the rollout of it through the North Shore Birding Festival Facebook page. All right, well, let's go on then, and I will introduce turn it over to Dr. Forsman. Thank you for coming.
Great, thank you so much, Deborah, for that introduction. And hello, everybody. I was going to say it's nice to see you, but I can't actually see you. But I am very excited to be here today to talk to you a little bit about all the exciting work that we're doing with Purple Martins at the University of Central Florida. Hopefully next time I'll be able to see you all in person. But for now, I am glad that we have the internet, so I can still connect with you this way. All right, so I thought I'd start out, since I haven't spoken to this group before, I thought I'd start out to tell you a little bit about myself. Deborah already told you that I'm at the University of Central Florida, but I'm not a Floridian originally. I'm actually from the country of Sweden. That's where I was born, and I spent much of my childhood going back and forth between Sweden and the United States. And thanks to very supportive parents, I developed a love for nature very early on, as you can see in this picture of me holding a toad. And so over the years, whether I was in Sweden or in the United States, I spent a lot of time outdoors and really cultivated a love for all animals and plants. That has hung with me over the years, stayed with me over the years, and I think I knew very early on that I wanted to be a biologist. So when I finished high school, I went on to the College of William and Mary for my undergraduate work. And this is really where I discovered my love for birds. You can see here the house wrens and the tree swallows, which I went on to study in my graduate work. But at William and Mary, I discovered my love for bird watching and being outside and field biology in general. And when I graduated from William & Mary, I knew I wanted to go on to graduate school. I knew I wanted to become an ornithologist, but I decided to take a few years off from school, and I worked at the Dartmouth Medical School. And this is where I discovered my interest in laboratory biology, specifically working with DNA and RNA and thinking about genetics and genomics, which I promise I'll define here in a little bit. So after Dartmouth, I went on to Illinois State University for my master's degree. I worked with Charlie Thompson there. He's been working on house wrens for a very, very long time, has a really beautiful nest box population of house wrens. And so I worked on extra pair paternity in house wrens. And so I brought in some of that genetics work that I had learned during my time at Dartmouth to try to answer questions about why house wrens and other species engage in extra pair paternity. After Illinois State, I went directly on to Cornell and the Lab of Ornithology there and worked with David Winkler to study tree swallows. And for this dissertation project, which I'd love to talk to you about another time, but for this project, I was very interested in how bacterial communities in the environment that tree swallows come into contact with influence their immune system function and development in offspring. So I finished that up in 2016, and then I ended up at UCF. And all of the work that I had done in the field, but specifically in the lab with genomics, is why I ended up at UCF. And I work on a lot of different systems here, but my two primary systems are purple martins, which we'll be talking about today, and also the Florida scrub jay. However, because of my interest in genomics and extensive experience with these types of methods, I also actually work on a whole suite of other systems too. I have a lot of work in sea turtles. I have a fantastic undergraduate student, Christine Sarkis, who's been working on a sea turtle project now for several years. Manatees, I collaborate with Dr. Anna Savage on amphibian projects. I have other collaborations with folks outside of UCF. We're just starting up a cottontail project. So lots of different species, and humans included. Earlier today, I was actually in the lab working on human samples. So humans and lab mice also are included in that list. So a great diversity of different systems, which makes it really exciting. But of course, my heart belongs with the birds. And so my two primary systems are the purple mart and the Florida scrub jay here at UCF. So at UCF, I work in the Department of Biology. Specifically, I run the genomics lab and teach undergraduate genomics, which is a hands-on course that's taught in this lab that I'm showing you the pictures of here. The students get really amazing experience with techniques that it's very unusual for undergrads to get a chance to do hands-on, like DNA sequencing. I'm also part of the UCF Genomics and Bioinformatics Cluster, which is a larger group that pulls from the Department of Biology, but also from biomedical sciences and computer sciences. So it's a really exciting place to be, lots of really exciting research. And so now I promised Deborah that I would define genomics. So I'll stop here to do that. So genomics is the study of the genome. And the genome is the collection of all the genetic material in one organism. So you have a genome, I have a genome, my cat has a genome, and that's the collection of all the genes, all the genetic material. So the study of genetics, generally we look at one or two genes at a time and how they're inherited. With genomics, we actually look at the entire collections of genetic material in an individual. How do we do this? We do this specifically by DNA sequencing. And over the last decade or two, this technology of DNA sequencing has advanced very, very rapidly and now allows us to sequence entire genomes, which is very, very exciting. So we can sequence the genes in the genome. We can also look at how these genes are expressed. And we can also sequence the genomes or parts of the genomes of other organisms that may be associated with us. So the microbiome, for example, the bacteria that live in our guts or on the skin, we can collect a sample and sequence a portion of their genome to figure out what bacteria are actually there. Since we can't actually see them, we can use their genetic material to do microbial forensics, basically. So genomics is super exciting. And for me specifically, my research, I like to think about DNA technology and genomics as a very powerful tool for addressing questions in ecology and evolutionary biology, specifically bird behavior and physiology. And so I would consider myself a molecular ecologist because I use molecular techniques, specifically DNA, to answer questions in ecology. So the students that are in my lab, they work on a whole suite of different types of projects, both with birds, but also with other organisms like sea turtles. But my own research interest in a nutshell is trying to understand how how microbial organisms in the environment, including parasites, influence the hosts that they're associated with. So, for example, how the gut microbiome in your gut influences your digestion, your health, your reproductive output, your senescence, and lifespan, and all those sorts of things. And again, specifically in birds. All right, so enough about me. Let's shift our focus now to the star of the show, the purple martin. Now you know a little bit of a background of my research interests, and you'll see that coming through now as we're talking about the purple martin and how we're addressing some of those questions in this wonderful system. So purple martins are swallows, which means that they belong to the family Hirundinidae, and there are about 80 species of swallows worldwide. About 29 of those species are in the New World. And so what I'm showing you here is actually a map of the distribution of the barn swallow, which is a cosmopolitan species, so very widespread. But again, there are a lot of species, and we have quite a few of them in the New World. So the purple martin takes first place for being the largest swallow in North America. And they are migratory birds, and so we see them here in North America in our spring and summertime. Here in Florida, it's more like winter and early spring. So they come up and breed in much of the United States and into Canada, and also parts of Mexico. And then in the fall, late summer, early fall, into later fall, further north, these birds start migrating down to South America, where they spend our winter before they return again the next spring. So the purple martin weighs about the same as a large kiwi fruit. And I will say here that I include this slide thanks to my son. This is his favorite slide of all the slides that I have about purple martins. And I actually think that it does a really good job of demonstrating how big these birds are. So the wingspan is about as long as the height of a bowling pin. So about 40 centimeters long, and they're about half that in length. And again, the weight is about that of a kiwi fruit, so between 45 and 60 grams, depending on age, of course, sex, and also subspecies. So purple martins are sexually dimorphic, which means that males and females look different. So the bird here on my right is a male. We know this bird is at least 2 years old, and this is a female over here. And the reason we know that this bird is at least 2 years old is because purple martins have delayed plumage maturation, which means that males during their first breeding season look more like females. And so it can be a little bit difficult to tell the females from the first year males apart. But after the males return for that second breeding season, they will have this very distinctive plumage. Purple martins are aerial insectivores, which means that they feed on the wing. And in fact, it seems like they like to feed at quite high altitudes. And so they fly around and hunt those bugs in the air. So they're very much dependent on flying insects, and so can be quite affected by bad weather that prevent those insects from flying around. We don't know tons about what purple martins eat, but we are hoping to change that here. So I will talk a little bit more in detail about that a little bit later on. So stay tuned for that. So purple martins lay between 3 and 6 eggs typically, and females incubate for a little over 2 weeks before those chicks are hatched. The nest cup is very shallow and is made up by in this case, pine straw or small sticks and straws, pieces of grass, and usually these birds also will line their nest with leaves, as you can see here. Seems like our Florida purple martins really love the live oak leaves. That's what we find most in our nests here at UCF. So after the incubation period, the chicks hatch out and they spend about a month in the nest. And so we have a couple pictures here that we took from this last field season. Down in the right-hand corner, we can see some chicks that just recently hatched out. These guys here are maybe about a week old, and these larger ones are about getting ready to fledge. After the birds fledge, they generally do tend to sleep or roost in the gourds for a little while after they have fledged. So we actually have 3 subspecies of purple martins. The ones that we have here on the East Coast are the eastern purple martin, the Subis subis. And these are the ones that we often see around these big colonies that folks will put up in their backyards and on the shores of lakes, and they really do like to be near water. So they are colonial birds. They like to nest in large groups like this. We'll come back to the eastern martin, since that's the subspecies that we work with here at UCF. But I also want to introduce you to the western purple martin, which is slightly larger than the eastern purple martin. And as the name implies, they live on the west coast. And these birds, there has been some success with getting them to nest in artificial nest boxes, but not as successful as on the East Coast. So, but there are definitely folks out there that are working hard to help these martins. One example of this is the Olympic Peninsula Audubon Society. And so they have a project where they're setting up houses for these western purple martins that seems to be very, very successful. So that's super exciting. And then finally, there's the desert purple martin. And these guys are so cool, I think. These birds actually nest in the saguaro cacti. And so they use old nesting cavities in these cacti. They're also colonial. As we can see here, a whole bunch of them checking out the saguaro and fighting over a nesting cavity there. So I just think these birds are amazing. There isn't a lot known about them yet, but that is changing. And so if you're interested in learning more, I really recommend that you check out the Tucson Audubon Society's website. They have some great information about the project that they're starting there with getting a better handle on the biology and natural history of this subspecies. And so there's some really great pictures there too. They've been using endoscopy actually to be able to get into these cavities and see what's going on inside. So if you're interested, I definitely suggest you check out the Tucson Audubon Society. So regardless of the species, the subspecies, purple martins are colonial, especially the eastern subspecies. As you can see here, they really are attracted to the breeding activity of other birds, of other purple martins. And so when folks set up breeding colonies like this, a new colony, oftentimes you'll put out a decoy or even use some playback to attract the birds to the chattering of other purple martins. And that seems to be very, very successful. So these are definitely gregarious birds. Traditionally, purple martins have used natural cavities, so those excavated by woodpeckers, for example, for nesting. So here we see both the western purple martin in a tree snag here, and then of course the desert purple martin in the saguaro cactus. So the eastern purple martins, though, are really interesting. They actually have quite a long history with humans, interactions with humans. And they now almost exclusively use human-made or artificial housing. There are only a few places left that we know of that these birds are still using natural cavities, and seemingly not in very large numbers. And so the fate of the eastern purple martin really is in the hands of folks like you and me that may be putting up these boxes in our backyards, because that's what these birds are so dependent on now. Interestingly, one of the last holdouts for these birds is our own Orlando Wetlands Park. And so we know that the birds have been using natural cavities here, and I can report that when I've been out bird watching this past spring, I did see a bunch of fledglings out there. So we are very hopeful that they are still using natural cavities out there. And so I just wanted to put a plug here that if there are any of you that spend any time out at the Orlando Wetlands Park and you're interested in helping us document natural nesting of these purple martins, please contact me. I'll leave my contact info on the very last slide. We would love to hear from you. We'd love to set up a network of folks to go out there on a regular basis to see if we can locate these birds. So as I said, purple martins have had a long history of relationship with humans, dating back to before Europeans came to the United States. And so there's documentation that indigenous peoples, especially in the southeast, were using gourds, hollowed out gourds, and setting up these gourds for the purple martins. And even today, we often will use the artificial cavities will be gourd-like. And some people actually still use the natural gourds as well. But many of the purple martin houses that are made now actually resemble gourds in appearance. So that relationship has been around for a long time, and many have speculated why indigenous peoples may have been setting up these houses. And so some ideas may be for natural pest management, although we do know that they don't tend to eat as many mosquitoes as perhaps we'd like for them to eat. But there are, of course, other agricultural pests that it might be helpful to have purple martins around, especially in larger numbers. And of course, as we can see, this is not a purple martin. Obviously, it's an oriole here that's mobbing a corvid. And so there are other types of pests other than insect pests, of course. So maybe that's one of the reasons. But another reason may just be that purple martins are really wonderful to have around. And they have a lovely chattering, and they don't mind to be around people. And so there's the aesthetic aspect, and perhaps even going back some cultural reasons that maybe they believe that they brought good luck. And they are in fact wonderful birds, and there are a lot of people around the country that appreciate them, and there are purple martin festivals happening every year, and so we definitely have a lot of fans of purple martins. And what's so wonderful about this, I think, is the very strong community-based science that has come out of this appreciation for the birds. And so purple martin landlording is a really popular pastime. There are thousands, if not tens of thousands of people around North America that set up these nesting sites for the birds and care for the birds and provide them with clean and safe places to nest every year. And of course, this also facilitates conservation and just general appreciation, which is wonderful. So I haven't mentioned it yet, but the Purple Martin Conservation Association is another wonderful organization that I recommend you checking out if you're interested in purple martins or purple martin landlording. And so the PMCA has been around for quite some time now, and over the last 25 years, they've accumulated over 1 million nest records from landlords across the country that have been submitting their data. So not only are these landlords providing safe places for the birds to live, but they're also contributing important data to science. So when and where do we see purple martins? As I mentioned earlier, we start seeing them here right after Christmas or around Christmas time, and they begin breeding in February, March, and once they are done, they pretty much head out to go south for the winter time. And so here is a map from the Purple Martin Conservation Association that shows just this. The first arrivals are here in Florida and up in Louisiana. And by January 15th, they are definitely here and then moving their way northwards. How do we know this? By observation. Again, community-based science has been really helpful here. Through the Purple Martin Conservation Association, there's opportunities for submitting data, and also eBird. I know a lot of us bird watchers use eBird, and that's also super valuable, contributes super valuable data. So the Purple Martin Conservation Association has a scout arrival study that they've been running for several decades now. And every year you can go and visit the map here that shows all of the arrivals across the range. So for 2020, just to give you an idea of how many people are involved with this, there were over 3,000 reports that were submitted to the PMCA. And over the last 22 years, it's been over 70,000. So lots of great data from citizen scientists all over North America. I just want to zoom in here so you can see what Central Florida looks like. And again, to put a plug in here that we need more data. And so if any of you out there are interested in contributing to this project, please contact me or contact PMCA so that your bird watching expertise can contribute to this great data set. Okay, so that's observational data. There are other ways of getting these data too, and one of those is by putting trackers on the birds to actually be able to follow them. And there are different types of trackers out there, and I'm not going to spend too much time talking about that today, but it allows us to see where and when these birds are moving. So in this case, not just arrival and departure dates, but actually seeing the paths that they're taking. Okay, so that's a little bit about Purple Martins in general. I want to shift gears now and tell you a little bit about my lab and also the Purple Martin Project at UCF and the things that we are doing and hope to do in the future. So I am not alone in this lab, thankfully. I have a great group of students. These are all undergraduate students that are in my lab or just recently graduated, but are still working on projects. And you may recognize some of these individuals because they are also Audubon folks. So Lauren, Brian, Julia, and Stephanie are all founding members of the Nighthawk Audubon chapter at UCF. Stephanie was the original president and Julia the vice president. And then last semester, Brian became vice president and Lauren president, and Brian has since graduated, and so, but Lauren is still our president. So we have a lot of exciting Audubon stuff happening at UCF as well. I have two students that are not working on birds. Victoria, a pre-vet student who recently graduated, is actually working on a gut microbiome project in mealworms. And then this is Christine, who I mentioned earlier, who is working on a project with sea turtles to use genetic methods to figure out what those sea turtles are eating. So great group of people. I couldn't do this without them. And although they all work on different projects, I didn't mention Isabel is a newcomer to our lab. She comes from the Audubon Raptor Center, the Audubon Center for Birds of Prey in Maitland. And she did an internship there this summer. And so she's now working on the Scrub Jay project with me to look at blood slide, lots and lots of blood slides to figure out how those birds are doing, and to look at parasites, potential parasites. So, these folks work on all different types of projects, but they have all been involved with the UCF Purple Martin Project to some degree or another. And so last fall, we received the good news that we received permission from UCF to put up Purple Martin houses across campus. So in February, we had received all of our polls, and we received really great advice from Dr. Jason Fisher with Disney Conservation and Joe Segrist, the president of the PMCA, to make sure that we picked the optimal sites for putting these houses up. And in February, we got all the students together, and over the span of a couple days, we put up 12 gourd racks and 144 gourds. We put all of them very close to water. Luckily on campus, we have quite a few ponds and smaller lakes. And so we could provide optimal habitat for these birds. Purple martins do like open habitat. They don't like for their housing to be very close to trees or shrubbery. And we also made sure that all of our poles had predator guards. And another thing that we did with our houses was to put pine straw into them, which is something that's something that's done by most, I would say, Purple Martin landlords, but we don't really know. We haven't really tested whether the birds actually have a preference for the pine straw or not. So that was something we wanted to do this first field season in spring 2020. And so we actually put pine straw on half of the gourds, and the other gourds we left without anything. So they were completely empty to see what the bird's preference would be. Like I said, we had a lot of help on the ground to get these poles up. And the birds like to have their houses raised quite a bit in the air. And so all of these houses have a hand crank so we can raise and lower the actual gourds. As you can see here, none of these folks are wearing masks. And so this was, in fact, back in February, before spring break, before we knew what was going to be happening. So it was a lovely sort of last hurrah, I guess, with all of the students to get out there and work as a team to get these poles up. And then we all left for spring break. And when we came back, the Purple Martins had found us, which was super exciting. Unfortunately, so did COVID, and campus shut down and was shut down for quite some time thereafter. And so our first field season was kind of a flop. And so because of that, I don't have a lot of data to show you from 2020. But I would love to tell you a little bit more about what we're planning going forward, and also some of the work that I'm doing with Purple Martin collaborators. But I can tell you that we had 12 nesting pairs and 50 fledglings. And so although I was not able to get on campus to do the typical things like banding and collecting morphometric measurements and so on, I was able to at least get to campus to check on when eggs were laid, when they were hatched, how many chicks there were, and fledging fate. So we were able to get some baseline data, and of course we were just happy that we were able to attract birds this first year, because we didn't know whether that was going to be the case or not. I can also tell you that the study that we did with the pine straw to see whether the birds preferred that or not, it was very, very obvious. Of those 12 successful nests, only one of them was in a gourd that did not have pine straw. And so starting this next field season, we will be putting pine straw in all of our nests. Well, so my students weren't able to come back to campus after spring break. And so I had to do the nest checks by myself. But some of the time I did have assistance from my 7-year-old son. So even more community-based science here. One thing that was super exciting too, I think, especially for the students, was that before spring break, UCF was able to come out and interview us and take lots of pictures while we were setting up these houses, and the PMCA then ran that story in their spring issue of the Purple Martin Update. So we were all very, very excited that we were able to get this project off the ground, and to have the great support of so many people, including UCF and PMCA. All right, so shift gears a little bit here again and tell you about the research that we hope to do going forward. And some of the research that I've been doing with collaborators that are also working on Purple Martins throughout North America. So some of the topics that I hope to address with the UCF Purple Martin Project going forward is looking at health and disease in these birds, foraging ecology, so again, thinking about what these birds are eating, genetics, of course, genetics and genomics, behavior, and including migration. So thinking about movement ecology, both on small scale and for dispersal, for example, and the larger scale, thinking about migration and changes in timing of migration over time, which is one of the interests of my current student, Lauren Puleo, who is the president of our Nighthawk chapter. And of course, conservation underlies all of this, especially my collaborative efforts with folks across North America and also into South America. So I want to take a moment to tell you a little bit about the work we're doing with foraging ecology. So basically, we're trying to figure out what these birds are eating, which can be very difficult to do, right? So we can observe what they're catching if those bugs are large enough. But oftentimes these birds are coming back to the nest and they have a bolus full of food in their mouth and you can't tell what they have. And so you could collect the fecal material and try to decipher what the birds have been eating by identifying insects on the other end. But of course, they've been digested and that can be really difficult to do. But as a molecular ecologist, I'm always thinking about how I can use DNA technology to answer questions. And so in my lab, we collect a lot of bird poop. We collect a lot of fecal samples from birds because we can answer a lot of different questions with that material from a molecular perspective. So let me explain how we use fecal material from birds, in this case a chicken, not a purple martin, to understand what these birds are eating. So a bird, a purple martin or a chicken, will of course take in a varied diet, in this case different insect species, a whole host of different species, which will move through the digestive system and become digested and come out on the other side as a fecal sample. Well, the great thing about these fecal samples is that they include lots and lots of DNA. Not only do they include DNA from the host, so the bird that's been sloughed off from the digestive system, but lots of DNA from those insects that the birds have eaten, and also any of the microbes that may be in the gut. And so from this, we can do fecal forensics. We can collect that DNA, extract that DNA, and sequence that DNA to figure out what the birds have been eating. We can also look at what bacterial species are present in the gut, and also if there have been any parasites. So there's lots of great information that we can learn about birds and other animals. It's not just limited to birds, of course, but we can learn a lot from this fecal DNA. So this is the technique that we have been working on in my lab now for the last year and a half. Once we collect those fecal samples, we bring them into the lab to process them, again, to extract that DNA, prepare the samples for sequencing, and then to sequence to get that genetic code. And so we're very fortunate at UCF to have our own DNA sequencer at the Genomics and Bioinformatics Cluster Lab. And so we're able to do all of this work in-house. My students are able to learn all of this, which is really great. That's part of the undergraduate genomics course that I teach too. It's amazing that the students can work with a sample and work through the entire process in the lab and then actually do the DNA sequencing and then the sequence analysis at UCF. It's really awesome. So that's the process for conducting a DNA metabarcoding experiment. We call it metabarcoding because we choose one portion of the genome to target that serves as a barcode that will allow us to identify the species that particular piece of DNA came from. And so it's just like if you go to the grocery store and you pick up 2 packages of mac and cheese from different manufacturers, And if you scan that barcode, you can identify that this one comes from one company and this one comes from the other. And we're doing the same thing, but with the DNA and with DNA sequencing, and then matching that sequence that we get to a reference database to figure out that, oh, well, this is from a crane fly as opposed to a dragonfly, for example. So we've been working on this for a while, as I mentioned, and I have a great team of collaborators working with me on this. Joe Seegers, of course, up in Erie, Pennsylvania, and he's been collecting samples, and Kevin Fraser up in the University of Manitoba has also been collecting samples. We've been collecting samples here in Orlando as well. And then we have Brandon Honig. He is up in Pittsburgh, and he is our bioinformatics specialist. So he has been working really hard on the sequence data that we've been producing to figure out who these insects really are. And then, of course, locally, here's Jason Fisher, who I work with very closely on setting up the UCF Purple Martin Project and collaborating on a whole bunch of different projects, including this diet project. And then, of course, Stephanie Gaspar, who recently graduated from UCF, and she has been working really hard on this project as well in the lab. So, and Stephanie, many of you may already know her. She's the president of the Kissimmee Valley Audubon chapter, and she was also the founding president of the Nighthawk chapter. So lots of Audubon connections. So in 2019, we collected about 150 samples from Orlando, Erie, and Manitoba. And so this is our initial set of samples that we're hoping to use these DNA metabarcoding techniques to characterize diet so that we can compare across a large geographic area to see what these birds are eating, and also to compare between nestlings and adults to see if there's a difference in what the adults are feeding the nestlings versus what the adults are eating themselves. So again, great group of people. I couldn't do it without them. So these samples are actually in my freezer right now at UCF. I am getting ready to sequence them in the next 2 weeks, but I can give you a little bit of a snapshot because we went ahead and processed a small subsample. By small, I mean small. It was 4 or 5 different samples from Purple Martins to see if our methods were working, our lab methods. And so we sequenced those data, and Brandon produced the results here that I can share with you. So these are some of the species that we're picking up with our DNA techniques. A lot of moths, surprisingly. Here we have the dark swordgrass moth, the plain gray hawk moth, the bent line carpet moth, a lot of crane flies also here, and midges. A lot of midges we're picking up. Here we have the tree squirrel botfly. We picked that up as well. More midges, a lacewing, and an ant, which I think is really interesting. And I'm very interested to see going forward when we sequence our larger sample, especially here in Florida, because there's been some documentation that Purple Martins will eat fire ants. And so I'm very curious to see if we're going to pick that up in our samples. Of course, those dragonflies, the Purple Martins love their dragonflies and damselflies, and more crane flies here. And finally, some beetles too. We're picking up some beetles and some bugs that you wouldn't necessarily think of as flying insects or spending a lot of time flying. So it'll be really interesting to see. We didn't pick up any mosquitoes, so we'll see from the larger sample set if we pick up any mosquitoes. So lots of great information. We are super excited about this technology, and I hope the next time that I see you, I'll get a chance to tell you what we found with our comparisons across the range of the Purple Martin. So another thing that's happening at UCF in the relatively near future, hopefully, is the establishment of a Motus tower. And so the Motus wildlife tracking system is the largest network of tracking towers that are driven by individuals, basically, or individual entities. It's a wonderful network. There are lots of these towers around, many of them in Florida, and they work by we tag the birds. So of course this is not a purple martin, but we tag the birds, and as the birds fly by these towers, they ping the towers, and the data are collected and sent to a central repository that is publicly available. So we are very excited about the possibility of setting up one of these Motus towers at UCF. It is in the works. However, we're not sure whether it'll be coming this next year or not. So, but quite a few of my collaborators are using Motus tags, and we are learning so much about their migratory behavior and where and how and when they're getting down to their wintering grounds in Brazil. So more to come on that. As I mentioned, there are tons of these Motus towers, and so the Motus towers are on 4 continents so far. There are almost 1,000 of them, and we have quite a few of them here in Florida too, and as you can see along the coast up into Georgia and South Carolina. I feel like these towers are especially powerful when you have belts. So this belt that you see going across Florida, because it allows you to capture those birds that are flying down, that are migrating down Florida here. So we are very excited to hopefully put a Motus tower in the Orlando area so that we can contribute to this, and so that we can also start tagging our own birds at UCF. The last thing that I wanted to share with you is something new here at UCF. And so we are partnering with the UCF Arboretum. So the student interns at the Arboretum are growing purple martin gourds. And so here's the whole team out, or at least part of the team, with their gourd setup that they built. And the picture here of these bottle gourds was taken just a day or 2 ago, and so they're growing quickly, and we're excited about harvesting them and drying them and making them into purple martin houses. So I'd love to share with you as this progresses how they end up turning out. So that is all that I have for today. I'm ending with a picture of the first birds that we spotted at UCF, and actually our first poll that we set up at UCF. I hope that I've convinced you that purple martins are awesome, and that we have a lot of exciting things happening at UCF. And if you are interested in setting up your own purple martin colony, or perhaps you already have one, I would love for you to get in touch with me. And my email address is here at the bottom, just anna.forsman@ucf.edu. I would love to hear from you. And hopefully we can get more folks involved with purple martins and conserving this wonderful species. They are quite common. However, the populations have been declining. I just heard recently that we've lost about 50% of our purple martins in Florida over the last 20 years. And so putting up these houses for them is really one way that we as citizen scientists can help this species out. And so if you're interested, please let me know. Check out the Purple Martin Conservation Association. Lots of great information there too. And with that, I'd be happy to answer any questions.
Great. And anybody, please put your questions in the chat. I could ask, I had heard that they cluster and they can detect the clusters as they're roosting, migrating south, I think, in South Florida on radar.
Yes.
Tell us about that.
Yeah, so I personally don't have a lot of experience with that, but that is definitely the case. And so when these birds are roosting at night, in the mornings when they all wake up and they leave the roost, there are so many birds thousands, if not millions of birds that you can actually see that on the Doppler radar. And so it appears on these radar maps as these donuts that are donuts of birds that are coming out. So you can definitely look at a migratory behavior on a large scale from these radar images. So you can see when they're on the move.
And you said that the first ones arrive in February, and there you see like a scout that comes first?
Yeah, so here in Florida, actually, it's not unusual that we see them around Christmas time.
Oh, Christmas time.
Yeah. So the first ones will come and they'll be spotted around Christmas time, and then you have a larger number of birds that are coming in January, and by February they are definitely around and checking out potential nesting sites. So yeah, in Florida it's much earlier, and then of course as you get further north, the season gets pushed later.
And then they're all gone by now. October.
Oh gosh, yeah, they're all gone by now. So our birds here, they were laying eggs in March, and of course this was the first year that we had birds, so it might be slightly different than from established colonies, might be a little bit later than established colonies, but our birds were gone here in May. And they hang out for a while, but then, by August, September, our birds are down in Brazil.
Mm-hmm. And do you know anything about what they do down there?
Oh, yeah. So this is really exciting, actually. There is a large group of folks that are working on figuring out what the birds are doing in the wintertime. So some of my collaborators that I mentioned earlier, Kevin Frazier and Joe Segrist and Jason Fisher, as part of the International Purple Martin Working Group. So they've been tagging birds now for quite some time to see where they're going. And they have taken several trips down to Brazil to actually locate the birds, locate their roosts. So that's what they do in the wintertime, is that they find optimal habitat for feeding, of course, for foraging. And then at night, they again establish these very, very large roosts with thousands upon thousands of birds. And so it's been a little bit of a process to actually locate where those roosts are, because they also move from year to year. And but they have been narrowing down and have, like I said, taken several trips down there now, and they're actually tagging the birds. They're able to catch them in the roosts and tag the birds there. So there's a great very small documentary that was put out from their last trip earlier this year, right before COVID kind of blew up. And it's on YouTube. I don't have the link on the top of my head, but Deborah, I'd be happy to share it with you if you would like to share it with your members too. It's amazing. And you can go in and actually see their expedition and see some amazing footage of these huge roosts, just enormous number of birds.
Is it like in the Pantanal region, or what part of South America is it?
So it's in Brazil.
Mm-hmm.
And I personally have never been to South America, so my geography, knowledge of geography in South America is very limited. But Manaus is the Amazon where they travel to.
Mm-hmm. Wow. Okay. And I've got a couple questions. How have you had any other birds? Let's see, I can put it on. Invading the nests?
That's a great question. No, luckily we have not. And so the type of nesting gourd that we purchased is supposedly starling proof. And I know at least at one of my locations on campus, we had starlings hanging out in the trees nearby, and they never bothered the houses. One bird that we really worry about is the house sparrow. They can be just awful, as my understanding from other sites, but we have not had a problem so far. I have not heard any house sparrows on campus, at least around our houses. So maybe that'll change in future years when they figure out that we have these nesting colonies up. But so far we have not had any problems, luckily.
So you have 144 gourds.
We do.
Is that like a minimum for a colony, a good colony?
Oh, no, no. So if you wanted to, you could put up a gourd rack with, 6, 18, 24 on just that one rack if you wanted to, and you could certainly attract birds. So we chose the number based on budget and also the number of suitable locations around campus. And so if we keep attracting birds, there's definitely the possibility of increasing the number of cavities. Like I said, we had 12 pairs. So out of 144, only 12 of those gourds were used this past year. So it'll probably be a while before we have to increase, but we have space on those gourd racks to increase the number of gourds if necessary.
Cool. And one more question here is, how did purple martins live before human created housing?
So they would live in natural cavities. So in tree snags and old woodpecker holes. They're still colonial though, but yeah, they use natural cavities, which we see, of course, it's much more common with the Western purple martins.
Because people log out all those snags, right?
Yes, a lot of those snags have definitely disappeared.
Mm-hmm. And someone asked, how close do they need to be to water? I know this is Lori, and she has a good yard for it, but maybe she needs to know.
So, I mean, you could put the house directly on the water if you wanted to. It doesn't have to be right by the water. There are definitely situations where you can attract purple martins where your house is not directly on the water, but I would say that it helps. I think they spend a lot of time foraging over the water. And so if you have access to water, that would definitely be the place you would want to put it. But I don't know what the minimum distance or the maximum distance. I will say though, maybe more importantly is making sure that you are not close to trees or shrubs. So I would say at least 20, even 40 feet away from the closest tree or shrub is definitely preferable. They want it open so that they have a good view over potential predators.
Okay. And I wonder how they interact with the other swallows.
So I am not sure how they are interacting here on the breeding grounds, but I will say that on the wintering grounds, it is not uncommon for different swallow species to roost together.
Mm-hmm.
I don't know whether they forage together, but they do roost together. So you will have mixed species flocks.
They kind of are right in between the cycle of the barn swallows here and the tree swallows, right?
Yeah, so I was just at the wetlands now this past weekend, and the barn swallows and a couple tree swallows were all flying together. And when I saw the purple martins out there in the spring, it was the barn swallows and the purple martins flying together. So it seemed like they were at least hanging out. But I don't know what their interactions are here on the breeding grounds. But again, on the wintering grounds, those mixed species flocks do happen, which is not uncommon for migratory birds in general.
So this is a call to birders to help with your observations out there. A lot of our members frequent Wetlands Park, and
yeah, we would love that. So Jason Fisher and I have been talking about this, that we would like to set up a program to make sure that that the wetlands are being surveyed on a regular basis and in a standardized way to see if we can figure out if the purple martins are using natural tree cavities there, and maybe even finding what tree snags they are using, so we can follow them going forward.
Okay.
So please contact me if you're interested.
So, and that was anna.forsman@ucf.edu?
Yep. And you can just do a Google search with my name and UCF, and my lab website will pop up, and there you can look through, also learn more about my students and the projects that they're doing, and you can learn more about the Purple Martin Project. We have lots of pictures about the process and of the birds, and a news section too when fun things happen for our students. So,
well, terrific. Thank you so much.
Absolutely. Glad to be here.
All right. And good night, everybody. Thanks for coming.
Thank you.